First of all, as a youngest speaker in this group, I want to say thanks to the organizers for giving me this opportunity. So the topic of the research that I want to share with you in this talk is assessment of the integrity of blood-retinal barrier in a mouse mimic of Oculodentodigital dysplasia-related glaucoma. So there's no conflict of interest with any of the authors. So first of all, what is Oculodentodigital dysplasia? We call it ODDD. So it's a rare human disease, and it has a similar phenotypes in the mice too. It's an autosomal dominant disease with the mutation in the gene GJA1. There are currently 63 known mutations in this gene, and this gene expresses gap junction connexin 43. ODDD will cause different types of ocular abnormalities, for example, microphthalmia, elevated intraocular pressure, and so on. Other symptoms of non-ocular abnormalities, for example, loss of tooth enamel. So, in the research by Gabriel et al. In 2011, human ODDD patients with retinal and optic nerve abnormalities were analyzed, and there were two patients. In the patient one, with the deletion mutation at position 41 to 44 in the extracellular domain of gene GJA1, it showed characteristic phenotype of ODDD with the ciliary body cysts and the split irises, and two new symptoms with retinal dysplasia and dysplastic optic nerve heads. In patient 2, with the substitution mutation in the leucine at position 11, in intracellular domain showed characteristic phenotypes of ODDD with retinal dysplasia and dysplastic optic nerve heads. So, me and my colleagues in Western University Biology Department started studying and doing a research to identify the progression of retinal degeneration in ODDD. For this purpose, we used a mutant mouse model of this disease with the similar ODDD phenotypes and glycine to serine substitution mutation at position 60. So, in this research, by using different methods, for example, autonometry, OCT, and postmortem H&E staining, what we found was that ODDD will cause structural aberrations starting in the anterior segment of the eye. For example, in the cornea, in the iris, in the ciliary body, and also it showed reduced anterior chamber width. In my research, I wanted to see, does ODDD and also does exogenous factors have any effect on posterior ocular abnormalities due to ODDD? So specifically, I was looking at blood-retinal barrier degeneration. So a bit of introduction. Retinal barrier consists of two barriers, inner barrier and outer barrier. So inner barrier has retinal capillary endothelial cells, and the outer barrier has retinal pigment epithelial cells coupled with connexin 43. It has a function in regulation of ion and protein flux and also maintaining eye position. Defect in retinal barrier can cause different retinal diseases, for example, diabetic retinopathy, which everyone's familiar with. So in my experimental design, I had 5 ODDD mice. I compared them with 4 wild-type mice, all of them age matched between 9 to 14 months, so they were a bit old. I was looking at 4 different tissues: liver, cerebrum, cerebellum, and the retina. I was looking at 3 biological replicates. So basically, we were getting 3 different samples for each specific tissue, and we were doing 3 technical replicates. So basically, we were doing our experiments for 3 times. In this experiment, I was using Evans blue dye, which is a azo dye and has a really high affinity for albumin. I used this to assess blood retinal barrier and blood-brain barrier permeability. So basically, the method of this research can be divided into 4 stages. In the first part, I intravenously injected Evans blue dye into the mice, and then we were waiting for 30 minutes to get a complete circulation of Evans blue dye in the mouse. After that, we euthanized the mouse in the cavern dioxide chamber, and then by using transcardial perfusion methods, we were draining all the Evans blue dye out of the circulation. Then we were going back to the lab and by it's a method we call it a small scale analysis. So we were using 1 milligram of tissue, we were homogenizing it in a formamide, and then we were incubating it for 24 hours at 70 degrees centigrade. By using this, all of the Evans blue dye that penetrated into the tissue were getting drained out of the tissue into the formamide. Then we were centrifuging our samples at 3,500 rotation per minute, and by using absorbance measurements of supernatants, we were collecting our data, and then by using ANOVA and a standard curve, we were analyzing our data. So basically, for the third part of the method, which is absorbance measurements, we use a spectrophotometer. So basically, a spectrophotometer is quantitatively measuring the transmission properties of a material as a function of wavelength. As you can see, Evans blue dye has an azo group. This azo group will cause Evans blue to have a maximum absorbance at 620 nanometer, and minimum absorbance is 740 nanometer. Basically, positioning of the samples in the spectrophotometer has the same degree of effect on both the 620 nanometer and 740 nanometer absorbances. So as we wanted to have a really high precision in our experiment, we use background subtracted absorbance of Evans blue dye for our data collection. Then, as we wanted to rule out the effect of weight in basically Evans blue dye penetration and blood-brain barrier and blood-retinal barrier permeability, we normalized all our data to 1 milligram of tissue samples. Then, as we wanted to rule out the effect of injection efficiency, which is both internal and external, we normalize all the absorbance values for the brain and the retina to the liver of the same mouse. Because basically the liver has a really high circulation of blood, so we decided that is the best way. Then, as we wanted to make our data more understandable, we try to convert all our background absorbance values to the concentration of the Evans blue dye that were penetrating into the tissues. For this purpose, we made a standard curve, and we got a really good R-square value with 0.998. So then we converted all our absorbance values to the concentration of Evans blue dye penetrated into the tissues. At the final part of the experiment, we compared liver values of Evans blue dye with the brain and the retina, and it shows that liver has a really significant higher concentration of Evans blue dye than does brain and retina. It confirmed the sensitivity of the assay. So by doing this, we found that our assay is perfect and it can work. Then, in the final part, we compared the values of Evans blue dye concentration in the brain and the retina of the wild-type mice with the OEEE mice. And no loss of blood-brain barrier or blood-retinal barrier integrity was observed. Basically, it shows that no evidence of degeneration in the barrier was shown. And also, brain and the retina didn't differ in Evans blue dye concentration assay. So, in the conclusion, basically in the first experiment that we did with my colleagues in Western University, we found that OEEE will cause a structural aberration starting from anterior segment of the eye. In my research, I found that exogenous effects don't have, there's no evidence of exogenous effects on posterior ocular structural aberrations, and specifically in the blood-retinal barrier. So basically, we confirmed the anterior origin of OEEE disease. What's the future work of this experiment is that basically by doing this experiment, we found conformation of size exclusion. Maybe the small molecules, ions that normally penetrate the barrier, may cross the barrier in a greater amount in OEED and will trigger the exogenous effects and trigger the microglial activation, which cause degeneration of the retina. But this needs to be researched further in the future. And at the end, I want to say thanks to the Department of Biology, Ophthalmology and Biology of Western University, my supervisor, Dr. Kathleen Hill, and the laboratory members, Eric and Anson. Thank you.
Okay, thank you. This paper is now open for discussion. Any questions?
So basically,
Really, I just missed.
the concentration?
Molecule.
Sorry?
Molecule.
Molecule of, I should look. But the molecule of Evans blue has a really is acting just like albumin because has a really high affinity for albumin, and basically it is used as a viability assay to see whether the tissue is is degenerated and has is working as a barrier or not. So I'm not exactly sure about the weight of the Evans blue dye, but it is used as a really assay for seeing whether it's working or not. Yeah. 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 Exactly. So. It was interesting because we expected that it should have an effect on connexin 43, but maybe the result is that maybe Evans blue dye. One of the reasons can be that the Evans blue dye maybe have a really high affinity for albumin, and maybe albumin is is not a molecule that is acting on connexin 43. So maybe like a smaller molecules going there and just making a degeneration. It should be like a work further. The animals? No. They are OBBB animals. So we had the genotyping of animals. We in previous research we had 35 animals. In this research we had 5. So. Well, in the wild type, no, they don't have. We use the control, yeah.
It's that right? And maybe you can comment.
Right.
Thank you.
So, as you can see, you have started a discussion over here. I was going to start giving out the certificates, but I'll try and rescue. So first and foremost, when you are looking at this ODD or oculodentodigital dysplasia, it's a very rare genetic disease, very, very rare. And it is characterized by, I believe, very small eyes and those kind of things.
Yeah, exactly, like microphenotypes.
Those are the phenotypes. When you go into genotype, the connexin becomes a big player in this disease, especially connexin 43, which is a gap junction, as I spoke today earlier. So in this disease, what happens is the gap junction activity, the gap junction intercellular communication activity actually increases. But interestingly, the number of gap junction channels, they decrease.
Decreases, yeah.
So if you ask me and what I found, I was thinking about it, trying to wrap my brain around this. It's a very complex disease. But we found that the number of connexin 43 channels decreases.
Yeah.
And as a result, the gap junction intercellular communication also goes down.
Goes down, yeah.
Here is that there's a compensatory factor. The number of channels going down, but the gap junction activity is going up.
Yeah.
So that's the first thing. The second is what we had found is when connexin goes down, it brings down the role of occludin and ZO1, the two tight junction proteins, which are, well, there's claudin 5, but those are two very important tight junction proteins that maintain the blood-retinal barriers. So in response to the query over there, I think in your case, you probably might wish to look into the connexin 43 aspect in this animal model that you have.
So basically, because the patients that we had and showed like a barrier degeneration had different mutations. So maybe that's the reason, because we use the mice with I have the slide here. So
while you pull up the slide, the mutations is also a very difficult one. There are 20 or 28 mutations
so
there.
There are like for OEEE, there are actually like 63 known mutations. So we use one of them, maybe in different
That may not be
mutations,
the
yeah.
one.
Yeah.
That's a possibility.
Yeah.
Well.